And we're back. The Big Skinny with Lou Basenese, and that guy on that screen before us was not skinny. Gotta love the new graphics coming in from my boy Dodd. Always keeping me on my toes. Never know what's next, and maybe there's another surprise this issue. But hey, I want to tell you what's next right now, and this is one of the more compelling biotech companies that I've come across. It's not by random that we are sitting here interviewing the CEO of Gain Therapeutics. This is actually a company that I've known for the better part of a year and a half to 2 years. I'm an individual shareholder. And to put it simply, they are developing drugs that could have a very meaningful impact on some of the most difficult to treat diseases that are in existence. I'm talking about things like Alzheimer's, Parkinson's, and other unmet medical needs and names of diseases that I can't properly pronounce. So it is with great pleasure that I get to welcome the CEO of Gain Therapeutics here to talk in more specifics. Gene, it is a pleasure to have you join the Big Skinny. Welcome.
Thank you so much, Lou, and I really appreciate you giving us some time to talk about Gain.
Yeah, no. Hey, where in the world is Gene today? That's what I want to know, because I'm a little bit jealous. So tell our audience.
So I'm in Milan for a board meeting. We are an international company, so we have operations in Barcelona, Milan, and Lugano. So Milan's an easy place and convenient for us to meet quarterly for a board meeting. So that's what I'm doing now.
I'm jealous as a fellow Italian. There's some other Italians around. I'm coming into the next board meeting if it's in Milan. But
okay, well, hey, so just by the way, Gene is actually short for Generoso, and Mack. I don't know how they got MACK out of whatever last name we immigrated over with from Italy, but
I love it. Well, we're here with Generoso and Luigi Basenese Terzo. So let's get into this and talk a little bit of biotech, mix things up. Bolognese and biotech, who thought they'd come together? Listen, Gene, I gave just kind of a brief overview. If I'm an average investor, I wander to your website, I read something called, Unfolding the Next Generation of Allosteric Small Molecule Therapies, and I'm like, all right, man, you go to sleep, which is, look, you're appealing to the scientific audience. So in layman's terms, tell us what you're doing, and then we'll go from there.
So we're trying to affect proteins in a way that can improve their function, and all proteins have a function in our bodies. So it's just a very scientific way of saying we want to improve the function of key proteins that are responsible for promoting cellular health, and that's what we're doing with our discovery platform.
Right, and so we bring up on the screen here, and I know this is available in your investor presentation for anyone that's listening to this on a replay later, is really this idea that a lot of diseases can be caused by mutations that impact how proteins are synthesized and then transmitted throughout the rest of the cells downstream. I like to think of this as because I'm a simple guy, the analogy is if you ever had a garden hose that got kinked and the water stops flowing.
That's incredible. That's exactly the metaphor I'm going to use. Because you can kink the hose, but that water still has to get out.
Correct.
And that's a really good metaphor.
So, well, I'll take credit for it from here on out. I don't accept payment in shares, but no. Look, this is how so proteins get misfolded through genetic mutation. And again, I want you to elaborate on this. This is my understanding, and then you kind of elucidate it more. Proteins get misfolded. There's downstream effects of that because those proteins give clues to the rest of the body on how it's supposed to function. And then it has all these kind of knock-on effects that cause disease. So if you can unfold that protein and restore normal function, is it possible to reverse a disease? Is it just restore, where you're at? How does that work?
Yeah. So let me get to the mechanism of what we're trying to achieve here with the biology. So what we're trying to do is what we developed is a drug we think will be disease modifying for Parkinson's. And just as you said, we're trying to get to the underlying biology to fix that. And the underlying biology here is misfolded proteins. We all have proteins. Our body transcribes proteins all day long, every day. About 30% of our protein transcription winds up getting defunct, misfolded, and degraded, maybe prematurely, but we can manage that because we're healthy individuals without gene mutation, without other comorbidities or other factors that are impairing our health. In patients with Parkinson's disease, there is an enzyme in the brain that is not functioning optimally. It's called glucocerebrosidase. It's a mouthful. It's
Wow,
called
I'm not saying that 3 times.
yeah, something might appear. It's called GCase for short. So it's the GCase enzyme. So this GCase enzyme is really important. It performs a lot of important functions in the cell. Many cellular compartments. A cell is composed of many cellular compartments, and they all need to be functioning. If one of them goes down, it's just like any other operational unit. If something
Right.
malfunctions, it's likely that the whole operational unit might be impaired by that. Well, that's what happens with neurodegenerative diseases. Something in the cell is failing, and it might be broad enough to cause multiple things to fail. And then that's cell death, and then you have neuronal death, and then you have malfunction.
Right. And so
So
you're coming in and developing drugs, small molecules, so pills that can be administered to reverse this misfolding, right? And then I
yes,
guess, yeah, so let me, because this is what I always focus on in biotech, right? Data is what it's all about, right? If you don't have good data, you can't survive as a biotech. It's not about going to find a customer. If you don't have good data, that, i.e., the drug isn't safe or doesn't work, so
that's right.
you're out of business. There's no arguing about that. So talk to us a bit about the early stage data and what's the
Yeah.
next data that we're looking for here to kind of give you those clues that, hey, we've got something here that could really be profound.
Yes, great. So where we are in development is we have taken our compound through to safety and target engagement validation. So what we've demonstrated so far is that our lead compound, which is right now called GT02287 or 287 for short.
Yeah, when
So
it becomes
287.
approved, you'll come up with a really hard name
Exactly.
to
Yeah. No, we're already
pronounce
doing
it.
that work. We're actually already doing that work, and naming a drug is not a trivial
Yeah.
exercise. We can cover that in another conversation, but it's not trivial, and we're doing it now. But where we've gotten to this point is that we've been able to demonstrate that the compound is safe and well tolerated, and it also is modulating or activating, increasing the activity of its target, which is GCase, that enzyme I talked to you about. It
Right.
is an enzyme that is implicated broadly in Parkinson's disease, and if you can increase its activity, it maintains the housekeeping that prevents Parkinson's symptoms.
And then in your animal models, right? Because I know, and then you studied the animal preclinical first. What was the impact when you activated this enzyme? How quick was the impact? Again, I'm not making
Yeah,
the
so this has turned every key opinion leader and um, it's turned every key opinion leader and expert in the Parkinson's field kind of like on their back heels. What we've seen in the preclinical animal models is that there is, when we model Parkinson's disease in these animals, and then we introduce GT02287, we see a dramatic recovery in their functional improvement within hours.
Wait, so
That,
I want to make sure everyone heard that. You're saying, again, full disclaimer, it's in animal models. That doesn't
yeah,
always
that's
it doesn't always translate. That's the risk we
correct.
know here, but that's the
Yeah.
precursor step before you get into humans. Within hours, you're seeing functional improvement in terms of
Yeah,
just physical response improvement to the
mice,
degenerative
mice,
disease.
within hours, mice are able to hang on to wires for longer periods of time. They're
Some
able,
mice in my apartment can do that too. So here in New York.
they can walk, they can do all these physical tests close to the control animals, the ones that are not impaired, with our drug within hours. And so that is a little bit jarring to the field because we're being cautioned by the key. So we rely on key opinion leaders and we rely
Right.
on experts in Parkinson's to help us understand and develop the strategy for developing the drug. So we share the attributes we see in the preclinical models with key opinion leaders. We say, what do you think? How should we treat patients with this? What do you think the right endpoints are? What do you think the right dosing duration is? We ask them about every aspect of trial design to
Yeah.
help enrich our clinical trials. They have all unanimously said you cannot expect a human being who's had Parkinson's for 3 to 5 years to experience the same functional improvement so quickly. You're going to have to dose a long time.
Yeah.
But our view on that is, okay, we can't promise that. We can't promise that functional improvement that quick. But also,
If it happens, it'll be
the
known.
world was flat until it was proved to be round.
That's a good one. Yeah, you're right. And no, look, I think that's the potential
And
here
it's
because
both.
if look, there's
It's
other
going.
biotechs I've been involved with where it perfectly translates from the animal models into humans. So if it happens here, it's a complete paradigm change. I do want to, before we run out of time, there's a question here that came in that I want to make sure that you can advance. Here from Matt Brown. Do you observe any downstream effects like inflammatory effects in healthy volunteers in the phase 1A?
Excellent question. Excellent question. So we believe there isn't it. We haven't characterized it fully yet. We need to do more work, but we have run inflammatory panels on our healthy volunteers, and we saw some very unique changes in the inflammatory patterns. We need to back that up with more data. We need to understand it a little bit more because we don't want to spitball to the general public. We want to understand it first before we present what the case is. But there is something that's going on that's making us question whether or not Parkinson's is caused or affects inflammation, and that is something that's debated in the field. And we think our drug might be able to tease that out. Actually,
Yeah, no, that's interesting. I mean, look, these are the things like you look at the development of CAR T therapies, right? Cytokine storm, where you broadly activated the whole immune system. You get these downstream effects that end up, in some cases, unfortunately killing patients. So these are the things that are critical. I want people to understand that there's a lot that goes into it. I love your more conservative stance here of saying, hey, we see things. We're not sure. We want to validate that before we start broadcasting it. Before we kind of wrap here and end with what we need to be focusing on as investors as kind of catalysts, we got another question for you. Did you get fast track from the FDA for the preclinical and phase 1 results? I don't remember seeing that, but just the question.
we haven't approached the FDA yet. We plan to do that on the back of the study that we're currently conducting in actual Parkinson's disease patients, where we can see when the functional improvement takes hold.
Yeah.
And then we can go to the regulatory agencies and say, this is what we're seeing from a mechanistic perspective. What can you do for us in order to what can we all do? Because
Right.
the FDA is engaged. Like, we all want to bring new therapies to Parkinson's patients. The wind is at our back from a regulatory perspective. We just need to show them that data.
Yeah.
Once we have something from this phase 1B study that we're
And then
currently,
when are we going to get the data? Is it a mid-year readout? So there's some interim data that we'll
we're
get
going to
in the
have
middle.
so we'll have initial read in the middle of this year. It's likely the end of June. So because we need this data to inform our phase 2 trial, the next trial that we're doing.
Right.
So we're going to be happy to share that with investors and potential partners to show our progress, and that will be in the middle of the year. Yeah,
Okay.
likely.
Well, listen, everyone, be paying attention. Gain Therapeutics is a stock to check out. Gene, I'd love to have you back because there's so much more we can get into here. I mean, you have the backing of the Michael J. Fox Foundation, a lot of other key opinion and policy influencers, obviously interested for the right reasons here because this could be a paradigm shifting treatment in terms of Parkinson's. But the other thing I want to make everyone aware of is that you have a platform for drug discovery that is AI-based, that is helping identify these proteins that are misfolded, that could be unfolded in unique ways. So we'll save that for next time in Italy, as long as you promise me that. We're coming live from Italy next time you come on.
Absolutely.
All right, Gene. Hey, enjoy the rest of your trip. Safe travels back, and everyone, be paying attention. Put Gain on your radar, and we will certainly keep it on ours here and keep you updated with everything going on.
Thank you so much. This was way more fun than most of the spots that I do. So thank you.
Woo! I like it, man. That's the best endorsement we can get. We try and have fun, but be informative too. So I'll chalk that up as a win, man. Thank you again.